Very warm welcome to this video. Dr. Nair Raza here. So today we are going to talk about the COVID super vaccine, and we will look into this issue whether there is a potential remedy for all the current and the future strains of the SARS-CoV-2 virus, and that the mutants that we are recently looking all around the world, whether there would be some form of vaccine which would be able to tackle all these mutants. So in this particular lecture we would be looking at this issue. So without further ado, let's dive and get started. Now before that, please, those of you who are watching my channel for the very first time, please do subscribe to my channel, and if you like my videos, a thumbs up would be fantastic, and do share it with your friends. Fine, so let's start. At the moment there are three types of vaccines which are being used all over the world for SARS-CoV-2 infection. The first one is the group of vaccines which are known as the messenger RNA vaccines. So these vaccines, they actually contain the messenger RNA in a nanoparticle which is injected into a human person, and that goes inside the cell, and then it sort of translate into the spike proteins of the SARS-CoV-2 virus, which then induces your immune system to produce antibodies which then would be able to attack the virus in case if it really infects you, and would be able to neutralize that. And at the moment, two messenger RNA vaccines are currently being used. One is the Pfizer vaccine, and the other one is the Moderna vaccine. Then we have got the DNA vaccine, which are also known as the viral vector vaccine, and there are three types of viral vector vaccine at the moment. The first one is the Oxford AstraZeneca vaccine, which is currently being used in the United Kingdom and some other countries of the world as well. Then we've got the Johnson & Johnson vaccine, though there have been some issues related to Johnson & Johnson vaccine, and the FDA in the United States of America has halted the Johnson & Johnson vaccine for the time being. And then we have got the Russian Sputnik 5, which is also a viral vector COVID vaccine. So what's special about these vaccines are that they are using an empty viral vector, which in most of the cases is an chimpanzee adenovirus, and then they put the nucleic material of this virus, which is made by recombinant DNA technology, and then they are injected into the human person. And again, at the same thing, these and then DNA is encodes for the messenger RNA, and the messenger RNA then codes for the spike proteins, and then it induces your immune system to produce antibodies against those spike proteins, which makes you sort of immune against the real virus in case if it infects you. Then the third type is the killed viral vaccine. So these types of vaccines use the actual virus, SARS-CoV-2 virus, but it is neutralized, it is killed. So they actually inject the dead virus into your body, and then that virus is recognized by the immune system, and the immune system then produces antibodies against it. So in case if a real alive virus infects you, then the immune system would be able to attack and neutralize that. And at the moment, the vaccines, which there are many killed viral vaccines, but the one which are a bit more famous than the others are the Sinovac from which is known as the Chinese vaccine, and the Covaxin, which is being used in India at the moment. So these are the three general types of COVID vaccines. Now, if you see, these vaccines are either targeting the spike proteins, or they are just targeting the whole virus, the shell of the virus, and then the immune system responds to that. So the efficacy of these vaccines, it varies. Like in case of the DNA vaccines, it has been reported somewhere between 65 to 72%. For the messenger RNA vaccines, it has been reported above 90%. And for the killed viral vaccines, again, different studies have reported different types of efficacy. Again, it can be somewhere above 50% to 70%. Again, the population attributes have been different from where these trials have been conducted, but nevertheless, the vaccines are efficacious in majority of the population. So these are the current vaccines. Now the problem with these vaccines is that as the mutant strains are coming up, there is an issue whether these vaccines are effective against those new strains or not. There has always been a concern that as new strains come up, these current vaccines would not be that much effective against that. Again, trials have been conducted against some of the common, mutant strains which are currently in circulation, like the UK strain. We have got the Brazilian strain, which is known as the B1 strain. The UK strain is known as the B117 strain, and the B1351, which is known as the South African strain. So then again, certain reports have said that probably the vaccine might not be effective against the South African or the Brazilian strain, and there have been a lot of concerns. And now, certain vaccine producing companies, they are doing trials on the new mutants as well. They're trying to alter their vaccines to see if they are effective against the current mutant strains or not. Now, the issue is that as long as the virus is in circulation, it's going on, like, every time it enters a human body, it undergoes a process of replications, like where millions and billions of viral copies are formed. Now, every time a virus replicates, there is always a probability of a change in the genetic material, which we call as mutation. And that mutation can lead to, not in all the cases, but sometimes it can lead to a more resistant strain. So as long as the infection occurs and the viral replication occurs and the infection goes on and on, there would be mutant strains which would be coming. So at the moment, we have got these three strains, which I told you earlier. We have got the UK strain, B117 strain, that's the other name for it. We have got the B1351, which is known as a South African variant, and we've got the P1, which is known as a Brazilian variant. And recently then we have got the Indian variant as well, which say probably they've got more than 13 probably mutations on the spike protein. So the thing is that with these mutants coming in, and probably there will be some more mutants which we don't know at this point in time, but probably future would show us that there are some more mutant strains as well, there would be a constant need to redeploy vaccines, like constantly redevelop vaccines to incorporate for these new mutants. Like, for example, in annual flu vaccine, every time each year a new flu vaccine is prepared and is administered to the populations who are at risk. And the reasons for having a new vaccine every year is that every year new strains that come up of the flu virus, their antigens are incorporated in that new vaccine. So flu vaccine is redeveloped every year. So there's a concern that whether we are moving in that direction where we would have to constantly redevelop these vaccines to incorporate for these current mutant strains, some of which are thought that probably the current vaccines are not that much effective, or about those mutant strains which would be coming in the future. We don't know about that. Like, what sort of mutants would that be? There's a possibility that these vaccines might be total failure against them. So then the question is there a way to develop a vaccine which is effective against all these strains? Now, theoretically, like, let's do a sort of a brain exercise. Is there a way in which we take care of all these current strains and maybe the future mutant strains? Now, if you look theoretically, for that we have to develop something which actually hits something in the virus particle which is constant. Like, if the spike proteins are changing, obviously you have to change your vaccine as well. But if something is constant again, in all these strains, then probably that can become a target for vaccine development, because in that case, if something is constant, that you can hit that and that would produce immunity, and that immunity would give you sort of a protection against all the strains, because that particular protein that you are targeting, that is constant. So that would be present in all the strains. So that is the underlying principle which is being looked into at the moment. And the National Academy of Sciences recently produced a paper which was published in this, what we call as the Proceedings of the National Academy of Sciences. And this article was titled "Hold Kill Genome Reduced Bacteria Surface Expressed Coronavirus Fusion Peptide Vaccines Protect Against Disease in Porcine Model." Now, this is quite an interesting paper and it's worth reading, because in this paper, what they have done, they have tried to target a protein which is known as coronavirus fusion peptide, And I will tell you what this protein is, because this is a very interesting protein. This is a protein which is constant in all the strains of coronavirus. Whether you are looking into the alpha coronaviruses, whether you're looking at the beta coronaviruses, or whether you're looking at the SARS-CoV-2. So there is one protein which is constant in all these strains, and that is the fusion peptide. So what they did, like they did some animal studies. So they took the SARS-CoV-2 virus and they developed immunity against this fusion peptide, and then they developed the vaccine based on this model, and then they injected them in pigs, and they found out that it protected the pigs against different types of coronaviruses which were causing different types of infection in the pigs. So they thought that probably this analogy can also be applied to the humans, where we can make a vaccine which targets this fusion peptide of this coronavirus, and then that would induce the immunity which would be effective against all types of coronaviruses. So the question is, are we looking at a super vaccine? A super vaccine is that vaccine which is effective against all these strains of coronavirus, whether you talk about the alpha coronaviruses, whether you talk about the beta coronaviruses, whether you talk about the SARS-CoV-2, or whether you talk about those viruses which cause diseases in the animals. Like there are variants of coronavirus which causes basically zoonotic infections. They cause problems, diseases in the animals. So that sort of vaccine which would be effective against all the strains would be known as a super vaccine. So how this super vaccine can be developed? Let's look into the SARS-CoV-2, its structure, and then probably you would understand how this is theoretically possible. And if it's theoretically possible, there is a sort of chance that it might be practically possible as well, because some studies have been done in pigs and they have shown promising results, as is shown in this particular paper. So let's study the SARS coronavirus and see what answer is given to us. Now, if you look at this is a rough diagram of the SARS-CoV-2 virus. So you can see this is a virus. It's got spike proteins on the surface. Now, if you magnify these spike proteins, and on this side I have magnified it. So if you see, each of this spike protein has got two arms at the end. So on one end you have got the proper spike protein, which is actually a protein made up of different amino acids folded onto one another, and this is the one that actually induces immunity by producing antibodies. Now basically all the mutant strains so far, they have got mutations over here. So the mutations cause the different amino acid and different protein structures over here, or the spike protein part. So what happened? The shape of the spike protein changes, just like a key, where if you change the shape of the key, so it might be easier for the key to lock into the ACE2 receptor. So most of the mutations, whether you talk about the B117 mutation, the B1351, the South African mutation, or the Indian mutation, or the Brazilian P1 state mutation, all of these mutations have occurred in this part, and that has led to change in the structure of the spike protein, making it more easier to fit into the ACE2 receptors. And when it is easy for this spike protein to attach to the ACE2 receptor, then it becomes easy for the virus to infect the cell, go inside, and start replication. The other arm of this spike protein is what we call as the fusion protein. Now scientists have seen, like the virologists, that this portion, this one over here, this one is constant in all the strains of SARS coronavirus. So whether you talk about the alpha coronaviruses, whether you talk about the beta coronaviruses, whether you talk about the animal coronaviruses, whether you talk about the SARS-CoV-2, this fusion protein has been constant. At least it's been constant, like in 93% of the SARS coronavirus species. So they thought, if we target this particular protein, because this is constant in all the species, then the immunity which would be produced against this fusion protein would be able to give you protection against all the variants of SARS coronavirus, those ones which are currently in circulation around the world, and maybe the future mutant strains as well. Because if so far the fusion protein has remained constant, then there is a high probability that in future also, in all the mutant strains that are going to come, most probably this fusion protein would remain constant. So this super vaccine, so-called super vaccine, which would induce antibodies against this particular part, this would help giving you immunity against all these strains. Now, what happens if you look here? When the SARS coronavirus comes and infects a surface epithelial cell, so there are receptors on the surface of the cell, especially in the lung epithelium, which we call the ACE2 receptor. So this spike protein portion of that, actually over here, this fits into the ACE2 receptor, and then this fusion protein actually fuses with the cell membrane, and then this fusion protein actually helps. Once the spike protein is locked in the ACE2 receptor, the fusion protein attached, which is known as fusion because it fuses with the cell membrane, and then what happens? It leads to the process where the whole virus is engulfed into the cell, so it goes inside the cell. So if we develop antibodies against the fusion proteins, what would happen? The antibodies would come and they would lock the fusion protein. So the spike protein is still free. So the spike protein would still go and attach to the ACE2 receptor. But because the fusion protein part has been blocked, this virus is not able to go inside the cell. And this is the principle. So you still are getting the SARS-CoV-2 virus getting attached to your ACE2 receptor, but because the fusion protein has been blocked, because now this end is blocked by the antibodies directed against that, so this virus is not able to go inside. And if this virus is not able to go inside, it's not able to cause infection. So what the scientists they did, they tried to produce this fusion peptide, and how they did is they took E. coli bacteria. E. coli has got different strains, the enterotoxigenic, enteropathogenic, enterohemorrhagic, etc., etc. So they took the E. coli bacteria, and the bacteria themselves have got like proteins on their surfaces, so they just like shaved off the proteins from the surface of the E. coli, and then they injected them with plasmids. So plasmids are basically genetically modified material which you can inject into the bacteria, and the bacteria would then produce proteins according to the genetic code of that plasmids, and those proteins would be then, they would appear on the surface of the bacteria. So what they did, they made special plasmids which had the genetic code of the fusion protein. So they injected that into the E. coli bacteria. So when this plasmid went inside the bacteria, it ordered the genetic material of the bacteria to produce fusion peptides, and these fusion peptides were expressed on the surface of the E. coli bacteria. So they cultured this fusion peptide of SARS coronavirus by injecting plasmids into the bacteria through recombinant DNA technology, and that led to these bacteria producing the SARS-CoV-2 fusion proteins on their surface. So you've got now a culture of bacteria which are producing SARS-CoV-2 fusion protein on their surface. So then they take these fusion proteins from the E. coli bacteria, they make a vaccine out of that, and they injected the pigs. So they injected that in the pigs, and they found out that it actually gave protection against the porcine diarrhea. The porcine diarrhea is actually caused by one of the animal strains of coronavirus. So these surface proteins, as I told you, these was extracted, a vaccine was made from them, and they were injected into the pigs. And then they found out that this not only gave immunity against the porcine diarrhea, porcine epidemic diarrhea, which is caused by a strain of SARS coronavirus, which causes diarrhea in pigs, but it also protected them against SARS coronavirus too. So they actually injected the SARS coronavirus in these pigs, and they found out that on one hand, it was protecting them against the epidemic diarrhea in pigs, which is caused by another strain of coronavirus, and it also protected them against the SARS coronavirus too. You see, both different strains, different genetic material on the spike proteins, but the fusion protein was similar, so it gave cross protection against two different strains. So this is how they did. They did this study in the pigs, and that was published in the proceedings of the National Academy of Sciences, and that has given a hope that the same thing can be replicated in humans. The same model can be used in humans to produce immunity against the existing strains of SARS coronavirus and the future strains of SARS coronavirus. So, in a nutshell, this has actually opened doors for the possibility of a super vaccine, and this super vaccine would be based on fusion peptide rather than producing antibodies or immunity against the spike protein. Because why? Because the fusion peptide is similar across 93 to 95% strains of coronaviruses, whether they are the alpha coronaviruses, whether they are beta coronaviruses, or whether they are SARS-CoV-2. So recombinant DNA technology can be used to produce fusion peptides of the SARS coronavirus, and then it can be injected in the form of a vaccination to induce immunity against fusion peptide. So if you are infected by any strain of coronavirus, whether it's the alpha coronavirus, whether it's the beta coronavirus or SARS-CoV-2, what would happen? The spike protein would still be able to attach, but because the fusion protein would be blocked, it would not go inside the cell and would not cause infection. And that would mean that this would be a super vaccine, which is probably the need of the day, as infections are still going on. Certain countries in South Asia are seeing huge infection rate, huge mortality, huge morbidity. So we might see more strains coming up. So that's why now the focus should be on development of a super vaccine. I hope you have understood in this small video lecture the concept of a super vaccine and how the scientists are trying to develop this super vaccine, and how they have tried to initiate this by doing some experiments in pigs, in animals, and are trying their level best to produce a super vaccine for the benefit of the humanity, which would be effective for people who are currently suffering from SARS, and even those people who might suffer in the future because of the mutant strains. So if you've got any questions regarding this, you can put it down in the comment section below, and I will try my level best to answer that. Thank you very much and have a good day. Bye bye.